Go ahead and get rolling through this. So, we left off last time with a question, and I left you with a cliffhanger. So, many cancer cells are resistant to apoptosis. So, what makes their mitochondria different? What's different about cancer cells that makes many of them resistant to apoptosis? And that is a good question. So let's just talk a little bit about what happens in cancer generally. And one of the things that does happen is you have rapid growth and proliferation of cells. And because you have these very metabolically active and rapidly growing cells that generally tend not to be because they are growing so quickly, the vascular supply can't keep up. Now it does to some degree, but it can't always completely keep up. And what this does is it leads to a lack of the normal things you would get delivered to a tissue, including oxygen. And this causes hypoxia. Now generally, long-term hypoxia is going to kill cells. So again, why doesn't it do it in this situation? Hold on a second. Get out of there for a second. Cell biology lecture Genetics 2016.
Did you take that picture?
No, I wish I did. I just realized there's a couple slides missing here. Okay, never mind. I'm okay. Okay. Move back to this. I thought I was missing a slide. I wasn't. Never mind. Okay, so why doesn't this long-term hypoxia kill cancer cells? There's a couple of reasons here. First off, cancer cells can do two things. First off, they're going to upregulate glycolysis. If you are no longer able to use aerobic means of respiration, then you fall back to using glycolysis and anaerobic metabolism that we've talked about already. One of the consequences of that is that it turns out that hexokinase will inhibit mitochondrial membrane permeabilization. So the more hexokinase, again, the first enzyme in glycolysis, will inhibit you from starting the beginning of apoptosis. So this makes their mitochondria more resistant to beginning the intrinsic apoptotic pathway. In addition, yes? Membrane or mitochondrial membrane permeabilization. So upregulate means in this case that you have more of the, in this case of this pathway, is that you're going to have more of the enzymes associated with that pathway. Or downregulate is going to mean that you have less. So we have that going on. Also, if you are predominantly using your anaerobic metabolism, you are not going to be using your mitochondria nearly as much. So mitochondria are predominantly going to be involved in you're not using them, if you have no final electron acceptors, you have no oxygen, you don't need to use those as much. It actually turns out that in lack of oxygen, mitochondria actually become damaging. Electron transport chain enzymes, backed up with electrons that they can't use, will start just dumping out those electrons into other things and begin causing all sorts of damage. So mitochondria, in the absence of oxygen, can become very damaging. So you don't want them as much. So there's a lot of pathways intrinsic that if a lot of cells undergoing long-term hypoxia will downregulate mitochondria, which is the same thing that will happen here. You have your mitochondria, you're dampening down your ability also to go through apoptosis, at least with the intrinsic pathway that we talked about already. So both of these things are conferring some degree of resistance to apoptosis to cancer cells. What questions do you have on that so far? Yeah, of course.
So because they're upregulating glycolysis, that means that they are downregulating other means of respiration?
It doesn't necessarily mean that you have to, but in this case, this is happening.
Is that as a result of, or?
No, both of these are a result of hypoxia. They're both a result of hypoxia. Because you upregulate glycolysis, you get more hexokinase, which causes a decrease in your ability. It inhibits mitochondrial membrane permeabilization. That specific enzyme inhibits that process. Also, having less mitochondria means that you're less able to go through apoptosis because that process largely happens in mitochondria, or at least is initialized in mitochondria.
So just like as a result of them rapidly growing because of some like mutation, then they have hypoxia and then they're even less likely to have apoptosis.
Yes. What other questions do you have? Yes.
So does that mean that like cancer cells are more likely to die if they have better blood flow?
In this particular case, they're more likely, if you have better blood flow, to undergo apoptosis for this one particular. This is one specific. This is one specific aspect of this, though, and a very, very multi-pronged. The cancer growth itself is very. There's a lot of factors coming into that. And this is just one factor. Does that make sense? Yeah, of course.
How does the hexokinase inhibit?
To be honest, I'm not very sure how it does that. In fact, I don't know if we specifically know how it does that. Very possible that we can find out. Any other questions? Yes.
You ended last class with here.
Oh, yes. So how are cancer cells different that they don't undergo apoptosis? Sorry, that's not very good cliffhanger. If you have to wonder what it was, by the time you come back. Yes.
When you say their mitochondria are downregulated, you mean like the activity, or like there are less?
There are less of them. They get recycled. You actually have mitochondria getting recycled on it. You have organelles. You have this thing called which essentially your cells will essentially digest organelles on a somewhat regular basis. And mitochondria, if you remember, this happens to be more often in mitochondria than other cells or in other organelles because, again, malfunctioning mitochondria are like horrible. The malfunctioning mitochondria mitochondria make tons of oxygen-free radicals, and then it's really damaging. It's all over the place. The electron transfer chain specifically is dealing with a lot of high energy, relatively high energy processes, and when they go wrong or start to go wrong, they start to do damage to things around them. And so, because it's just, they're very reactive. And so, yeah, you turn over the mitochondria relatively rapidly. And so if you're producing less, if you're turning over less, and you just stop producing them at quite the same rate that you have been, then the overall number can go down very easily. Yeah. What other questions do you have? Yeah. Yes. Yeah, inversion. That's all you were inversion.
So like both of these responses, I guess, or both of these mechanisms are intrinsically meant to be protected for something else, right?
Yes.
And then we can't, like cancer cells just like use them to go rogue, basically.
Yes. Yeah. So under normal circumstances, these are going to help cells long-term deal with hypoxia better, but in this case, they have to deal with hypoxia because they have already done rogue, and it makes them inadvertently more resistant to apoptosis. That makes sense? Yes.
So, like, I just, like, it seems, because, like, if you have, like, if you're increasing your glycolysis, like, if you would want to, like, increase your membrane permeability, right? So, like, you would.
If you were taking the pyruvic and moving it into there, yes. I mean, well, not necessarily the permeability itself. So remember the mitochondrial membrane permeabilization is the first step of apoptosis. And when you do that, it stops all of the functioning of the mitochondria because your protons leak out and the cyclone C is leaking out, and that totally stops everything. So in the case of this, like, you don't necessarily want to do that. You don't want well, let me back up. In the case of increasing glycolysis, under normal circumstances, you don't want to do that because they don't need to be anything in or out of there. But also, you don't want to blow up your mitochondria in a way that's going to kill the cell. In which case, if you're going through membrane or mitochondrial membrane permeabilization, that's leading to cell death. You don't necessarily want to do that just by upregulating glycolysis.
So it doesn't, yeah, so, like, it just, like, goes through its channels and stuff, and it doesn't matter, like,
Well, pyruvate is going to be mediated specifically by channels that bring it in. Yeah, it's not going to be mediated by the permeabilization that's happening because of bad backs and those things. Make sense? Any other questions? In that case, let's talk about our next thing, photosynthesis. So, in photosynthesis, I'm going to give you a very brief overview of how this works. First off, just the general concept here is that we're going to have light energizing and exciting some electrons. Light hits it, you're excited, and these electrons are going to move along an electron transfer chain that is therefore going to pump protons across a membrane and cause a proton motive force. This is very similar to what we saw happen in mitochondria. And then ATP are going to be generated by that proton motive force. We are also going to generate this other molecule known as NADPH. It's going to be formed from NADP+, and is going to accept some electrons. So NADPH is somewhat similar to NADH, as you probably inferred because of its name. The difference here is we do have the same basic structure, is that we have a nicotinamide, we have adenine is a dinucleotide. The big difference is this ribose that's on the adenine has 2 phosphates attached to it. One to a phosphate that's just kind of by itself, and then the other that has the phosphate attaching to the other nucleotide as well. So we have an additional phosphate on NADPH. So, in addition to this, a couple things just from a brief overview. In the difference from electron transport chain that's happening in your mitochondria, where the electron source is coming from oxidizing glucose, or actually in that case pyruvate in the Krebs cycle, is going to come from water, H2O. We're going to split water and use the electrons that we get from that to do this. Yes?
So what about animals that photosynthesize?
Animals that photosynthesize are just swiping things from plants to photosynthesize, or other photosynthetic organisms to synthesize. So they're not really doing it any particularly better or different.
So then the organisms that they're swiping from, they still take in water that the animal consumes?
Yes, they're going to use water in some place at some point. Yes, this is all going to come from water. So and it's all going to happen in a chloroplast. Whatever is photosynthesizing, this is all happening in chloroplast or a chloroplast-like thing. So some animals that you might be thinking of that photosynthesize could be like tridacna clams or Elysia chlorotica, the sea slug that does that. Essentially all they're doing is they're using what was made by other organisms to do that. In the case of Elysia chlorotica, they eat algae, and as they eat the algae, they separate out the chloroplasts from the rest of the algae that they're eating, and they place that into their tissues, and it photosynthesizes, and they swipe the stuff from it. There's a lot of other really cool things going on there, but yeah, that's the basic gist of that. In case of like other things like anemones, like Anthoplura or Tridacna clams or other stuff like this, they actually live symbiotically with unicellular algae in their cells that have their own chloroplasts and just running through doing like kind of the normal thing, and they're exchanging nutrients with those. So at that point, it's not really the animals photosynthesizing, so to speak, but a symbiotic relationship, or what's known as kleptoplasty, where they're stealing it from something else that's doing that. Does that make sense? Okay. Yeah.
You said the pyruvate is the electron source?
Well, the electron transport chain, like once we talk about Krebs cycle, yeah, it's pyruvate that's feeding into that's doing that electron in our electron transport chain going to Krebs cycle. That being said, the ultimate source of that is from glucose or whatever else you ate, depending on which pathway it's coming. Does that make sense? Yeah. So, the ATP and the NADPH are then used, once we have those generated, are going to use to fix CO2 into pre-existing organic molecules. So we're essentially going to take pre-existing organic molecule, shove another carbon on there from the carbon dioxide, call it good. And this is what's known as CO2 fixation. This process is going to take highly oxidized CO2. It's going to add it to organic molecules in the cell. They're going to be reduced by NADPH, donating electrons and thus reducing the CO2. And the energy to do that is going to be provided by the ATP. overall, this is kind of your overall basic reaction that is going to happen. If we take the things that we start with, the things that we end with in this. Now, this isn't exactly stoichiometrically correct, but approximately correct. Is that we're going to take 6 carbon dioxide, and in the process of doing that, use 6 ATP and 12 NADPH and 12 waters. Using these 6 CO2s, we are going to get 1 molecule of a hexose, generally glucose. And we're left with our 18 ATP have been hydrolyzed into ADP plus phosphate, and then our ADPH has turned into ADP plus some protons. So let's begin to talk about the specific places in which this is happening, the chloroplasts. So the chloroplasts are going to be slightly different than the mitochondria, in which we talked about things similar to this happening before, which have 2 membranes in 2 different spaces. The chloroplasts have 3 membranes in 3 spaces. It's like the plants said, we can do it better. We'll add 1 more on. I imagine if we ever find aliens that want to do it better than us, they will have 4 membranes in 4 spaces. And we'll feel wholly inadequate and be sad. But until that happens, chloroplasts are the only things that we have to worry about, making us feel inadequate. So generally, this is the general setup. We're going to go through each of these parts in turn, but essentially on this we have a membrane here, we have another membrane right there, and then we have these other sets of membranes in here. So let's begin to talk about each of those in turn. Like in the mitochondria, we have the outermost membrane. And also like in the mitochondria, it's very permeable. We have a porin-like protein in the outer membrane here, and this means that the general composition inside this membrane is going to be relatively pretty close to the same on the outside of this membrane. However, in the membrane, we have several enzymes for lipid synthesis here. So again, that is then inside there we have an intermembrane space between that membrane and the inner membrane. So that is this representing here, this very thin space between these 2 membranes here. Whoa, I'm not very stable. Inside there we have the inner membrane. Now, somewhat different than the inner membrane of the mitochondria, or very similar to the membrane of the mitochondria, this is also very impermeable, like it is as well. However, we do have a plethora of carriers and transporters and things so that we can selectively get what we want across this membrane. In this particular right here, I am illustrating there in red the inner membrane. Here, I'll point it out to you. Right there. Inner membrane. On the inside of the inner membrane, in the mitochondria, we call that the matrix. In this case, is known as the stroma. The stroma is now this space that is sitting in between the inner membrane and the thylakoid membranes that we're going to be talking about in a few moments. And here we have a lot of enzymes that are associated with CO2 fixation. We have starch grains, so these are storage of carbohydrates that have been made. We have DNA, RNA, ribosomes. Here's where most of the functionality for maintaining the chloroplast is going to be coming from. In this illustration, stroma is this area through here. So in this illustration, most of the area is stroma. On the inside of that, we have the thylakoid membranes. So thylakoid membranes are these small, flattened, pouch-like sacs that are sitting on the inside of the stroma. And these stacks of thylakoids are going to be called granite, or a single is a granite. I'm just wondering aloud to myself what the name for a group of them would be. Anyways, well, stack of ones called around. What's that?
Could you go back to stroma for a second?
So, one gram is one stack.
Yeah, one stack. So, thylakoid, 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 thylakoid. So this here is a granum. If I have more of these together are the grana. Is that? Yeah. Granite. My grandma always used to bake me cookies. Wait, what?
So the grana is a stack of
It's just more than one of the stacks.
Okay.
Plural for stacks. It's like stack and
So
stacks.
grana is one stack.
Yeah. Grana is one stack. Is this really confusing? I'm not going to ask you about the plural of this. That's not actually exceptionally important. It's just so that if I refer to them later, you know what I'm talking about. And if I start talking about my grandma, you know that I'm not talking about this. Okay, so chloroplasts in general, like mitochondria, their membranes have a lot of protein. So most of the membranes, I'm not going to distinguish between them, are predominantly protein, about 60% protein, and we have approximately 40% lipid. Of the lipids, that breakdown is about 10 to 30 phospholipids to glycolipids. Or if we're talking as a percent, I'm not going to make that. Now the membrane is very fluid. This is because there's a high amount of unsaturated fatty acids. And it allows for high mobility of the proteins through there. Now, if you remember with the fatty acids, that noise disturbs me. I feel like it's going to fall. And you have one saturated and you have one unsaturated. The more unsaturated fatty acid tails that you have, the less it allows the membrane to pack. And so, for instance, you have a high degree of like saturated fatty acids. Like this, the membrane can pack more tightly and it becomes less fluid. If you have more of these, then, I mean, this is kind of an illustration. Yeah, that's really very good. Excuse me. Like that, the membrane can't pack as tightly and then it becomes more fluid. Does that make sense? Yeah. Okay. So having a higher proportion of unsaturated fatty acids makes the membrane more fluid, and this allows proteins to move through that membrane more easily. So this also contains, especially on the inner membrane, or the thylakoid membrane, excuse me, pigments and some electron transport chain components. Let's take a look at that. This is, okay, so these are the thylakoid membranes of here. This and this and this. This whole thing would be a granum. All of these together are the grana. Now, inside the thylakoid membrane, we have this area known as the thylakoid lumen. This is the area inside the thylakoid membrane, and some of the electron transport chain components are inside there. There's here on the inside. Okay. So this whole scenario is going to get started with light interacting with some pigments. So let's begin talking about. So there's a whole list of photosynthetic pigments, including chlorophylls, carotenoids, and other pigments. The three primary ones are the chlorophylls. They look a little something like this. Many of them have magnesium at one end and then a long chain. I'm not going to ask you specifically about their structure. But specifically, they're going to well, maybe I have that backwards. Okay. So inside here is a lumen, and this membrane is this. Although this 5 and 6 are not supposed to necessarily line up with like this 5 and 6. I'm going through like, see, that's 3 there, and that's 4 there. Like this is like the numbers of the things I'm listing. So I'm talking first about the outer membrane, then I'm talking about the inner membrane space, then I'm talking about the inner membrane. Wait. What's that? I did not mean for them to be lined up. the point was to talk about them in order from outsidemost to insidemost. The point was not to line them up necessarily with this figure. Unless you want me to go through 12 points on it. Okay. So this, yeah, I think, so Doug Lumen, that's that right there. The numbers here are not meant to line up with the numbers I have on my slides, yeah.
So when we were talking about like the membrane, like all the membrane facts, is that just like in general, or is that the thylakoid membrane?
Okay, this is actually the thylakoid membrane. This is specifically referring to the thylakoid membrane. That being said, this is approximately the same as the inner and the outer membrane as well. I probably said that in a confusing way when I was talking about this. That's my bad. Oh well. Okay. Back to the photosynthetic pigments for a bit. Okay, so these photosynthetic pigments are going to absorb light energy, and an electron is going to become excited. It's going to go to a higher orbital at that point. That's very unstable for it to remain there. And so it can only remain there for about 1/100000th of a second, and then it has to do something. And so there's one of three things that can possibly happen once that happens. First off, you can have fluorescence. And this actually does happen relatively often with photosynthetic pigments. The energy that is in the electron can then get released again as heat and light. And generally the light that is released, specifically in fluorescence, is going to be at a different wavelength than was absorbed. And the electron returns to the original orbital. Alternatively, the electron can get transferred to another molecule. such as a neighboring pigment. Okay. So, with this in mind, let's begin talking about what happens during photosynthesis. Now, I'll put this up on each model. I'll take this image and put this up on there. So, it's going to start with, very briefly, it's electron transfer chain. But it's all going to start here with photosystem 2, the first in the chain, and that is going to be interacting with light and beginning the process of actually getting electrons that we are going to be passing on to this electron transfer chain. So let's talk a little bit about that. Photosystem 2 is going to contain lots of pigments, individual pigments. So somewhere between 200 and 500 individual pigment molecules are going to be a part of photosystem 2. They're going to be attached 3 to 8 polypeptides. They're generally the same pigment, like the same, like chlorophyll A, chlorophyll B, something like that, except for one of them, which we'll come back to. Yes? So, yes, it is, and that's because there are an insane number of different kinds of chloroplasts that have slightly different photosystem tubes. And so, yeah, it all depends on exactly what pigment that they're using, which we're going to talk about one specific one here, but also how this electron transfer is set up. So, really, this is like around a really huge, diverse range of organisms have photosystem tube. And everything from like huge trees, like imagine like a really large sycamore tree that you would find outside, and then think about red algae that you would find growing like in a hot spring. They're both photosynthetic organisms, but they're insanely different. Like we think of them as being, or actually here, even better, it's like you think of like the red sea that you can find on the ocean, like on the side of the ocean, right? It looks leafy and red. And then you have like something like a sycamore tree. You think in general they're basically kind of the same. They're photosynthetic, or you can just kind of sit there. But in truth, they're insanely different. They are insanely different. They are more different from each other than you are from a portobello mushroom that you would have on your sandwich. Like you and the portobello mushroom are more similar to each other than a red algae and a sycamore tree. Like by a decent measure. So when we're talking about, but they're both using essentially this same system for photosynthesis to some degree. But those same systems are radically different. Like, I mean, not radically different in that they're using this different basic pieces, but those basic pieces can be quite different. Does that make sense?
So the variance is more between different organisms.
Yeah. Yeah. The same organism is going to tend to have the same number of pigments, roughly the same number of pigments in its co-reactions. Okay, we'll leave it there and we'll pick this up on Wednesday. We are here on Wednesday.